I'm Michelle Marinkovich, the Associate Vice Provost for Undergraduate Education and the Director of the Center for Teaching and Learning. And thank you so much for coming to what is the last of the talks in the 2012-13 schedule. Not the last of the talks in the series, of course. It's just for this year, so stay tuned. We're very fortunate to have Professor Martha Cyert of Biology, who is also the Senior Associate Vice Provost for Undergraduate Education, and the Thomas W. and Susan B. University Fellow in Undergraduate Education, speaking to us on learning how to think like a scientist. This is an important topic and one that Martha has given a great deal of thought to during her time at VPUE, as she's provided leadership to undergraduate research programs and also to the Leland Scholar Program, a summer transition experience for Stanford freshmen from under-resourced high schools. I also have no doubt that it's a question that she will continue to address when she soon leaves VPUE to go back to concentrating more exclusively again on her beloved lab work. Martha showed an early, though by no means exclusive, inclination towards science in earning a bachelor's degree, summa cum laude, from Harvard in biochemical sciences in 1980. She went on to complete a PhD in genetics at UCSF in 1988 and then did a 4-year postdoc in the Department of Molecular and Cell Biology in UC Berkeley. After the postdoc, she crossed the bay to Stanford, where she has fortunately since spent her career, beginning as an assistant professor in 1992, becoming associate in 1999, and full professor in 2006, all in our biology department. Since her research is at the cutting edge of cellular biology, I cannot presume to explain it to you. Though I know, since Martha is an award-winning teacher, as well as a distinguished researcher, that if she and I had had the time to sit down and talk about her research, she would have made it both clear and compelling. Her work has been recognized by NSF's Young Investigator Award, by Procter & Gamble's Young Investigator Award, and Stanford's Terman Fellowship, among other honors. Her teaching has also been recognized by the H&S Dean's Award for Distinguished Teaching, and as I noted at the beginning, by the Thomas W. and Susan B. Ford University Fellowship in Undergraduate Education, a coveted honor. As you'll hear, she's made particular contributions to the curriculum of Bio 42 and 44X, the latter, the core molecular biology lab experience for our undergraduates. All of us who work with Martha are also struck by her fervent support for a more equitable and more diverse higher education landscape. She has devoted attention and effective advocacy to such programs on campus, including, as I mentioned, establishing the Leland Scholars Experience and creating with Tommy Woon a faculty group to support first-generation low-income students. Another of her creations, the VPUE Faculty Scholars Program for recently tenured faculty on campus, is likely to start a whole new phase in the careers and in the place of teaching in the professorial life at Stanford. For these and many other reasons, including the big ideas she is so good at generating and exciting us all with, it's a true pleasure to introduce Martha to you, and it has been a true pleasure to work with her in VPUE these last 3 years. Professor Cyert.
Thank you so much, Michelle, for that introduction, which is definitely appropriately embarrassing, and lets everyone know how old I am and how long I've been here. So thank you all for coming. And I have to say, it is intimidating to give a talk about teaching with people like Robin Dunbar and Michelle in the room. And I really can't presume to sort of tell you all the literature that's out there about how teaching in the sciences should be done. But what I decided to do instead is just really give you much more of my personal view, sort of highlighting the different courses that I've taught in my life and what some of my guiding principles are and really, where those have come from. So I'm going to start just very briefly, way back at the beginning, because I think, all of us, we've been students ourselves, and when you end up being a teacher, you definitely are influenced by the people that taught you. And it took me a long time to sort of think about who had been my major influences, and unfortunately never really got a chance to tell them how important they had been to me. So I'm going to tell you guys instead. Actually, can we turn the lights down a little? Because I don't know if you can see the pictures very well. This woman her name's Dorothy Cyert. She lived to be 98 years old. She was my high school biology teacher in 1974. This picture is about 10 years before that. I went to an all-girls school, so even though these students are from the 60s, we would have looked pretty much the same. We wore uniforms. And, why was Dorothy such a great biology teacher? I can't tell you a single thing, a fact that I actually learned from her. But what I will always, always, always remember is she just had this amazing enthusiasm. So she made biology fun. She made biology the kind of experience where, you go out in the sandbox and play and discover things. And really, I think, you guys can all go away right now. Because I think that's the most important thing. If you love your subject and you can impart that passion to your students, that's way more important than any kind of, factual information you're going to tell them for sure. So being sort of propelled, and I wanted to say one other thing about Dorothy, which is she was a close personal friend of Rachel Carson. So I grew up in Pittsburgh, and Rachel Carson is a very well-known environmental biologist and was also from Pittsburgh. And she and Dorothy had been close friends in college. And so a lot of what I remember about Dorothy, too, is she told us all these stories about what she and Rachel used to do in college and how they made cosmetics and sold them to all the other students and all that stuff. So she just was a big personality and influenced a lot of other students besides me. So then I got to college, not to Stanford, but to that other university in Cambridge, Massachusetts. And this man, David Dressler, really had the biggest impact on having me decide what I wanted to study. He taught a large introductory course about molecular biology. His name's David Dressler. And what I learned from him was how exciting it is as a student to really hear science unravel the way it does when you actually are doing research. So rather than being presented with a series of facts, David's lectures would basically set the stage for an important experiment that had been done, and he would literally walk you through that experiment. He would say, well, this was their question, and then these are the results they got. And you'd be sitting there thinking, oh my gosh, what do those results mean? What did they learn? What did they do next? And there was always sort of an appropriate gap in time before he then said, so then this is what it told them, and this is what they did next. And I will also never forget that engagement, that intellectual engagement, which is really the most fun thing about doing science. So I've tried to incorporate both of these role models in my own teaching. And as many of you know, especially Wahida, who's been partner in crime in Bio 42 for a long time, I actually taught the introductory cell biology lectures in this course for an embarrassing number of years, almost 20 years. And why did I start doing that? Because I showed up in 1992 as a young assistant professor, and my department chair, Bob Simone, said, you should teach this course in cell biology. It'll be really good for your career. So I said, okay. But then I got into it. And I think for a lot of faculty, the prospect of teaching in this large required introductory science course would kind of seem like their heart would sink. Like, I'd way rather be teaching, seniors who are really interested in my particular specialty. But I stuck with this course for so many years because I actually really grew to love the experience. And there are a number of reasons for that. And for one thing, it's the audience and your chance to have a huge impact. When you have a room full of, 200 to 300 students, you can, even if you influence just a handful of students in that room, I think that's a big impact. Also, it's really the first time that students at Stanford have had this topic of cell biology at the college level. And knowing from my own kids, their experience through school, I think in high school, some people luckily avoid this, but I think unfortunately in general, biology is taught in kind of a boring way in high school, and it's really taught as facts. And what my kids knew about cell biology came from, building models of the cell, just not very intellectual. So I really treasured the fact that I had the experience year after year of really being able to introduce students to thinking about what goes on inside your cells in a very different way, and a way that really relies a lot on chemistry, which is why we make them all take chemistry in their freshman year before they take Bio 42. Also, this course is a series, 41, 42, 43. In 3 quarters, students get really a whirlwind introduction to basically every important topic in biology. So that also means that there are particular challenges in Bio 42, which are, you just can't tell them everything in a particular topic. And it means that every time you get in front of them to give a lecture, you really have to make critical choices about what you're going to tell them, and most importantly, what you're not going to tell them. And it really forces you to strip the concepts down to figure out what really has the highest priority, what's the most important concept that you really want those students to get and to take away. And unfortunately, I also came to realize we all take this for granted, but in biology, there's just so much nomenclature. And there really is a lot of memorization that students have to do just to remember the names of proteins and whatnot. So anything, I just always tried to do what I could to minimize that aspect of the course and make sure that what was really important, that they knew what was really important was understanding not what the protein name was, but what it did, what its function was, and how the whole process worked. So now that I have you as a captive audience, and I know a lot of you are not biologists, I can't resist. Oh, sorry, I was going to say this too. And so really, I sort of developed my guiding principles, which have carried me through my whole time at Stanford from this first teaching assignment in Bio 42, which was, what do I really want students to walk away with? Especially in Bio 42, I think, one of the big goals is you want them to come back and want to take more biology courses. So the primary goal was really getting them excited about biology as a science and about the fact that biology really lets you understand how living things work. And I also really wanted to start introducing them at that very early stage to how science works. So especially from high school, students are used to reading a textbook and sort of memorizing the facts. So to really start getting them to question, where does that material come from? Why are these facts? Do we really accept them? I mean, in many cases, what I really like to point out was that something that was in their textbook in the current edition was then going to be quite different in the next edition because new experiments had come to light that really challenged the existing model in the field. So really trying to get across the point that biology is a science, and it's a constantly changing series of information, and even the most basic fact that you read in a textbook, you should be thinking critically about and wanting to know where that information comes from. So I just wanted to point out a couple things from Bio 42 that I really was surprised to find that year after year the students really loved the most. But in retrospect, of course, it makes perfect sense. So one of the lectures I had to give was about muscle contraction. And honestly, when I looked at the material the first time back in 1993 and saw that I was going to have to teach about muscle contraction, I just thought, oh, that's the most boring subject on the planet. How am I going to teach that? And of course, I didn't know anything about it, and I had only heard a few lectures about it, and the lectures I heard were boring. But over the years, what I realized was that every time I gave that lecture, the most number of students would come down afterwards, and they were so excited because the lecture really explained the chemical and biochemical basis of a process that we're all so familiar about. I was up there showing them this movie, which just ran, and for some of you, you know exactly what this is. This is showing you the biochemistry behind this process that happens when you move your muscle. And basically, how much cooler could it be than having students come in, listen to your lecture for an hour, and go away knowing next time when they move their arm or their leg, all the different biochemical processes that are going on inside your cells to make that happen. And this whole idea that actual proteins in your cell can combine in ways to do, manual physical work. So the sort of lesson that I learned from this experience in particular is that, of course, this makes total sense. The way to really engage students is to get them thinking about something they think they know a lot about, right? They have personal experience with moving their muscles, and yet engage them in the fact that maybe they've never really thought about how that process that they've taken for granted for years, how it really happens. So I find that really fun to be able to explain things like that to students. The other example I want to give from Bio 42 has to do with that goal of introducing students to think about where the facts they're learning in their textbook came from. So I always made a point, I tried in every lecture to at least tell them about one experiment that was done and how that experiment really contributed to major concepts in the field. And one of my favorite examples is this classic experiment. This is just a cartoon of the experiment from the Alberts textbook. But this is an experiment that I myself read about when I was a graduate student working on the cell cycle, and it really influenced me a lot. And what I love about this experiment is simple, and yet it told us a lot. And even if you have absolutely no technical background whatsoever, you can actually think about and appreciate the impact of this experiment. See, Wahida is nodding and agreeing with me back there. So here's the basic experiment. The question that these researchers, Rao and Johnson, were trying to answer is about the cell cycle. So as cells divide, they have to duplicate their DNA. And you know that the DNA is in chromosomes, and those chromosomes have to segregate completely accurately between the two sister cells. That's a process we call mitosis. And then after mitosis, cells go on into a growth phase, and basically every single cell can go through this whole series of events. So the question was, how is the timing of those events so precisely regulated? So you could imagine that if a cell tries to start dividing its chromosomes before all the DNA has segregated, that would be bad, right? And so these experiments were done in the 60s, and really no one had any idea whatsoever how those basic decisions were made inside a cell. So what they did was to take cells that were in mitosis. I don't know if you can see this, but these are little chromosomes here. And they artificially fused those cells with cells that were in other parts of the cell cycle. G1 is a growth phase. S is the phase when cells duplicate their DNA, synthesis. And then G2 at the end is another growth phase of the cell. And what they found is that when they fused a mitotic cell to any other type of cell, this mitotic cell always induced the other cell nucleus, this is a nucleus, these nuclei all then underwent the physical processes associated with mitosis, the most obvious of which was chromosome condensation. So I know that you guys aren't all biologists. What does that tell you about this mitotic cell? If you can fuse it to any other type of cell and now induce mitosis to occur, what would you conclude from that? I'm going to call on someone if no one volunteers. Oh, come on, just give it a go. This is the fun part. Someone who has never heard about this experiment before. Okay.
Some chemical in the mitotic cell that's causing it to undergo mitosis.
Exactly. And so that's so simple, and yet how profound is that? Because as soon as you know, wow, there's something in those mitotic cells that are going to make the other cell go into mitosis, of course, the next question is, what is that thing? And, 40 years later, we have a Nobel Prize given out for the scientists who really figured out exactly what those proteins are and those critical regulators. So that was just a little mini example of what I tried to do in Bio 42. And for one goal was really to introduce students to how science is done. But the other goal is to really get them to appreciate that science and research is actually a really creative endeavor. I think a lot of people that have not done research themselves have just sort of a general view that scientists are very logical and they're always analyzing processes. And yet, a lot of the classic experiments I got to talk about in Bio 42 were just beautiful examples of people that had the vision to even figure out how to start addressing a fundamental question in biology. And, that element of creativity, I think to this day is really what's the most fun about doing biology and about doing research. So I tried to get that across to them. Oops, sorry, I forgot to turn off my little updates there. That means people in my lab are working while I'm here, which is great. Updating their manuscripts on... Okay, so the next phase of my life, while I was still teaching Bio 42 actually, from 2004 to 2006, I joined forces with Tim Stearns, a professor, a colleague in my department. And Tim had written a grant to the Howard Hughes Medical Institute and had gotten a really nice fat grant, lots of money, to institute what he was pitching as a pre-graduate school curriculum. So once he got the money, he had to figure out actually what to do with it. And fortunately, I got to join with him in that experience. And what came out of that was that he and I together designed a series of courses called Bio 54 and 55. On the right, you'll see an ad that we placed in the daily. Students had to actually apply to get into these courses, and they were limited. We could only accept 16 students. And the whole purpose of 54 and 55 was, again, to really introduce students directly to that thrill of doing research. And this was geared towards sophomores. So these were mostly students that did not have a lot of research experience themselves yet. And as you can see, the way we enticed them was by saying, are you ready for an out-of-the-ordinary science experience? Do you want to design your own research project? And that was really one of the keys to Bio 54 and 55. I'll tell you a little bit more in the next slide about one of the projects that came out of that course. But first, I just want to emphasize what this is. So we taught this course for 3 years. We had 3 groups of just absolutely awesome, amazing students. Many of them did go on to graduate school. And there just was always an amazing energy and excitement and a lot of fun associated with the course. And this is the t-shirt that one group of students produced. And it says on the back of the shirt, top 12 ways you know Bio 55 has taken over your life. And I won't go through every single one, but some of my favorites are number 6, you're not surprised to see the entire class in lab at 2 a.m. on a Wednesday. And this was true. These students were so excited about what they were doing. We taught in the Clark Center. There used to be a teaching space there. That's been eliminated now. And they'd just be in there. They'd be having a party at 2 a.m. doing their yeast experiments, which was totally awesome. And then the other one that's funny, which probably a lot of people won't get, but number 2, a night of streaking and scoring doesn't appeal to you. And that has to do with the yeast genetics they were doing. Streaking means separating the yeast cells out to get single colonies. And scoring means looking at your plate and figuring out whether things grew or not. So it was a super fun experience and just really intellectually engaging. And I would have to say that my top 2 lessons that I learned from that whole experience was, first of all, definitely teaching is way more fun when you collaborate. And I was really fortunate to be able to teach with Tim in this course. And we've since gone on and taught a lot of things together, including the lab course I'll tell you about. And really that, I had never done that before. And I think big surprise, just like when you do science, collaborations are one of my favorite things. And having somebody to bounce ideas off of and challenge the way you think about things is just always great, no matter what you're doing. But then also, both Tim and I really got to see firsthand that, why were the students in there at 2 a.m.? They were there because they were doing their own project. And it was their data and it was their question that they'd come up with that they wanted to investigate during the course. And I won't go through the details about how that process happened. We did guide them towards developing these project ideas. But that really had a big influence on both of us. And again, it makes sense for those of us who are scientists and get to do our own research. That feeling of ownership and the fact that you get to decide what you're going to study and why is really a key motivating factor. So I just wanted to give you a brief idea of what kinds of experiments the students actually did in that course. So there were a lot of different projects. I'm just going to take you briefly through one to give you the general idea of what we did there. What we did was to use yeast. Yeast is a microbe. It grows really easily. It's really easy for beginning scientists, students, to learn the techniques that they need to learn to be able to do experiments with yeast. So we worked with yeast, and every year the questions that the students addressed had to do with natural products and how those natural products worked. So in some cases, the natural products were things associated with toxicity. Like one year we used the compound, one of the major compounds in Teflon, the non-stick surfaces. And you guys might know there was a lot of, especially a few years ago, a lot of stories in the popular press about, birds dying because people's pets' birds dying. Does anyone know about this? I'm getting blank looks. Because the compound from the Teflon would get vaporized as people were cooking. And it is quite toxic, and your body has no way to break it down. So that was one example. We used those products. But this example has to do with another compound called curcumin. And curcumin is this yellow powder. You can buy it in the health food store because it's actually been touted for many, many years, since 1900 BC, as a traditional medicine. So it's the major component of the spice turmeric. And if you look in the literature, you'll basically find claims that curcumin cures everything, from malaria to cancer to arthritis, digestive problems, you name it, Alzheimer's, anything you can think of. There's at least one paper saying that curcumin is a remedy. And actually there are clinical trials going on right now determining whether it does have a clinical effect as an anti-cancer drug. So the basic question that our students got to address was, okay, what does this thing do? How does it work? What does it do to cells? And what we learned was that if you took students, especially students that were willing to work till 2 a.m. in the middle of the week, and put them in the Clark Center and gave them some yeast strains and some media with curcumin, that they could use this tool to transfer a series of yeast strains, each one of which was missing one particular gene in the genome. Okay, so we had thousands of these mutant strains, and ordinarily people do these kinds of experiments with robots. We had undergraduates, and we had lots of enthusiastic undergraduates, and actually that was another lesson we learned, is that you know, in science, you really don't believe anything unless the results have been replicated. We had enough students that we could have some overlap and multiple pairs of students testing the same strains. So they would just physically transfer each of these strains onto media that contained curcumin. And what we were after was finding out which strains, particular strains, so again, each one of these, here's wild type strains, so this one's normal. All the other ones on here are missing one particular key gene. And as you can see, in the media containing curcumin, all of these strains showed really poor growth compared to the wild type. Each one of these spots comes from transferring a little drop of liquid culture that contains yeast cells onto the media. And so then you incubate the plates for a couple days, and when the spots all fill in like this, it shows you that all the strains are growing. Here they are growing without curcumin just fine. Here they are with curcumin, and they're all sensitive. These are the strains that our students identified by screening through that whole entire collection of thousands of mutant strains. So most of the strains did not have a growth defect on curcumin. This was just a specific subset. And then what was really interesting was that what you can see over here is if you provide not only curcumin in the media, but you also supplement it with iron, now all those strains are growing just fine, even though there's curcumin there. So basically what we were able to learn was that the strains that were all really sensitive to curcumin were strains that had a problem acquiring iron. So we then turned that into a hypothesis that what we thought curcumin was doing inside the cell was chelating iron, and that was how it was inhibiting the growth. And we basically proved that by showing that now they can grow when you give them extra iron. And then the significance of that in terms of understanding how curcumin might have its anti-cancer effects, for example, is that it may actually be inhibiting the growth of cancer cells by chelating iron inside those cells. And the chemists had already published several papers showing that this chemical does have the ability in a solution to chelate iron. So the other thing we learned from this whole experience is it is actually possible to publish a research paper based on findings that started with an undergraduate lab course. So that's something that was an important revelation. So after Tim and I got through teaching that course, so basically his grant ran out, and also there were really great things about teaching that class. It was really fun. The students were engaged. However, there were some downsides too, and one of the downsides was we really didn't reach very many students. It was a very labor-intensive course, and the students that took it all really had great experiences, and we've kept in touch with them. But we were only able to do this for 16 students a year. And meanwhile, in the biology department, we teach large introductory lab courses that hundreds of students take every year. And Bob Simoni, our chair, really challenged Tim and me to see if we could redesign those large introductory lab courses to incorporate some of the exciting elements that we had been able to offer students in this small boutique course. So this has been quite a challenge. And we've worked on it. Let's see, we had 2 pilot years. We had a year of just trying to figure out what we wanted to do. We had 2 years of pilot courses. And now we've had the full course running and offered for all students for the last 2 years, right? Okay. And let me just set the stage a little bit. So here are the courses. This is the one that Tim and I worked on, Bio 44X, and I'm going to tell you about that one. But at the same time, Tad Fukami, who's sitting right there, spearheaded efforts to redesign the other introductory lab course, 44Y, and you guys should definitely have Tad come and give one of these courses, one of these lectures, and he can tell you about his experiences there. He's also published a paper that is really gives you some insight into how the the strategies he took in designing that course. But I'm going to be telling you about 44X. And again, you know, you definitely can't take on any of these types of projects on your own. So we've had a great, great team working on this whole effort. And it's included me and Tim, Tad for 44Y, Daria Hekmat-Scaife, I'll show a picture at the end, Pat Sewell, who's here, and Shamala Maladi, who is not on this list. I'm so sorry. I don't know why you're there. Okay. Yeah, this is that list of authors is for the pilot. And Pat helped us get a grant from NSF to fund part of this. It was also funded by a Hoagland Award from VPUE, from CTL, and also from monies from the School of H&S. So before we redesigned the course, there was a perfectly good course that had been running for many years under capable direction of Shamala Maladi. But it's what we would describe as a classic sort of cookbook type lab course. The students had four independent units, and each of those units ran for 2 weeks. And each time they had a particular unit, for example, one unit would be called molecular biology. They would come in for each of the 2 weeks. They would follow a series of protocols. And the thing that we really wanted to change was in this format, really they were always doing experiments where the outcome was known and the students knew basically what the outcome should be. So what the good part of the course was that every 2 weeks the students were doing something different, so they got exposed to different organisms and different kinds of techniques. But what they weren't getting was that thrill of intellectual discovery, because they always basically knew what the outcomes of their experiments should be. And in fact, when the experiments didn't come out that way for some reason, students would be frustrated. They'd be sort of angry and be asking, well, why didn't it work? It clearly wasn't my fault. It must have been something the TA did. They didn't give me the right thing. They didn't explain things to me. You know, it was really sort of exactly the opposite of the experience that you have when you're doing research in a lab. So our goals were to somehow create a series of experiments that could be done by hundreds of students and yet would create more of an authentic research experience where students had that same feeling of ownership over their data. And what we really, really wanted to do, I mean, when Tim and I got together and talked about this was really our key goal, was that we didn't care about students' exposure to different organisms and different kinds of questions. What we cared most about was we wanted them to come away with the intellectual experience of what it was like to do research and to really emphasize those skills of critical thinking, data analysis, and communication of scientific results. And our rationale is that if you can hook students on the ability of science to basically answer questions, which is what we're doing here, that experience, even though maybe they've only been exposed to one type of experiment, that experience, if they really like it, will then drive them to go out and find other types of research experiences. And we're very fortunate at Stanford that undergraduates have a lot of funding and a lot of different labs available here in the medical school to then be exposed to different types of research. So we fundamentally changed the course. And instead of having a series of different experiments that they did every 2 weeks, we had students working on one research project that starts in week 1 and continues and builds through the whole course. So just to quickly give you an idea of what that project is like, we also knew that most of the students that take this course are interested in going to medical school. And, you know, thinking back to that experience I was telling you about where students are thinking about how their muscles work, and it's like, oh, cool, okay, that's something I want to know I want to know about. We similarly decided that a good way to hook students here would be to have them working on something relevant to cancer. Everybody kind of knows what cancer is. Unfortunately, many of us in this room have had either personal experience with cancer or someone in our family that's had cancer. So it's really sort of a natural way to grab students' attention and get them engaged. So the experiments all focus on a protein called P53, which it turns out is one of the major proteins that is mutated in cancer cells, in tumors. And the basic structure of the course was we had students working in pairs and carrying out this single project during the whole quarter. And each student, each pair of students in a room would be working on one particular mutation in P53. These are mutations always that have been identified in a cancer cell. And this picture here shows you the first thing they do, which is to figure out where in the structure of P53 their mutation lies. And through the course of the quarter, they're basically trying to figure out how that mutation, if that mutation disrupts the activity of this important P53 protein, and if so, how. And what we did to make this technically much, much easier, we again had the experience of having undergraduates work with yeast cells, and they pick up the techniques they need to know very quickly. It's very easy to work with. It's relatively inexpensive compared to working with animal cells. So Daria Heckmott-Scaife spent time engineering yeast strains to actually introduce either the wild-type P53 protein or one of these mutants. And it turns out you can assay some of the basic functions of this protein when it's expressed in the yeast cell. Yeast do not express their own copy of P53, so we're really using yeast as sort of a living test tube here. So I told you that really one of our primary goals was to emphasize the critical thinking skills. One of the things we did was to introduce a really simple mnemonic, and I use this almost daily now with people in my lab, with students that come to me to work on their honors theses, everyone. This is called the query method. It was actually developed by a graduate student in Tim's lab years ago, Charlie Anderson. And it's just kind of basic. Query stands for QU, question. Every time you describe your scientific experiment, you should start by describing the question that the experiment is trying to answer. Experiment. Next, you want to describe how the experiment works. Results. Next, you want to talk about what the results of the experiments were. And then finally, why is your interpretation. So we really introduced this and reinforced it over and over again in terms of the students presenting or thinking about or describing what the results of their experiments in the lab had been, all in this format. And the important thing is that they always have to think about the question. They always have to think about why they're doing the experiment. What's the goal? What's the issue that's being addressed? So overall for the course, the question they're trying to answer is what's wrong with your P53 mutant? Excuse me. So each pair of students has their own mutant that they're working on. So that gets to the sort of ownership aspect of the course. First, they ask where's the mutation located. Secondly, they ask whether it can activate transcription. I'm not going to go through the technical details here, but that's what the P53 protein does inside cells. Then they ask whether the protein is actually being expressed, because if it's not able to activate transcription, which many of the mutants that we've chosen to have them study have a defect. So then the question is, what could explain that defect. And the simplest way to explain it is if the mutant protein just gets destroyed inside the cell, right? So they have to actually do a technique called an immunoblot to figure out if the mutant protein is actually being expressed. One of the things P53 does is to bind a specific sequence in DNA and then activate gene expression. So they do an assay to see whether their mutation is affecting DNA binding. And then finally, these are yeast cells, and they do an experiment to see if their mutant protein can actually get into the nucleus. Because if the mutation was interfering with that, then even if the protein worked, if it can't get into the nucleus, that's where the DNA is, so it can't express genes. So I've already mentioned this, the way the course is organized overall. In each lab room, there's a single pair of students working on a particular mutant. But again, we have the advantage here of having hundreds of students take this course. So that means that there are multiple lab sections meeting either on the same day or different days. So we take advantage of that by having each student periodically during the quarter, students get together in what we call the mutant groups, where they come and talk about their results. So for those discussions, they're not getting together with the other kids in their lab room who are all working on different mutants. Instead, they're getting together with other students from other lab days or other lab rooms who are working on the same mutant that they're working on. And so they're like little mini research conferences where they get to see whether their results have agreed with the results of others, and often, if not always, the answer is no, they didn't agree. So then we have these really fruitful discussions about why they didn't agree, or maybe there was a problem, which has also happened more often than not, so that nobody's experiments really worked very well, and we really take the opportunity in these sections to talk about some of these issues, troubleshooting. Do we believe the data at all? Why should we believe it or not believe it? Students have to start thinking about the way the experiment was structured, positive controls, negative controls, all that. So these really become, authentic discussions about scientific results and allow students to grapple with really basic, important issues in being a scientist. So that's one way in which we're able to take advantage of the large enrollment. And then the whole course culminates with this just totally awesome poster session. This is a standard format for how scientists communicate their results at research conferences. We have our students do this, and we organize each row by mutant. So students are going around and looking at the posters from people that studied the same mutant that they did and students that studied a different mutant. And really the room is just abuzz with noise and excitement and discussion the whole time. So lessons learned. Teamwork, teamwork, teamwork. This was just a huge organizational feat, which would never, ever, ever have been successful without everybody's help. And especially, I want to point out Shamala, who's here, and Pat, who's here, and Daria, who's not here. And here are also some of the instructors from the course. Vina has moved on to another position, and Sarah Brownell, because I'm going to talk about Sarah in a minute. So really, I just can't even go into all the things that needed to be organized and coordinated to pull this off. And not shown here is the fact that we also have a large number of graduate student TAs working in each of these rooms, and some of our TAs from last quarter are here, so they can tell you about that. The other take-home message, and this is really where it's embarrassing to have Michelle and Robin in the room. In 20 years of being at Stanford, this is the first time I've really had an actual scientific assessment of the course I'm teaching done. And man, it's great. You have all these ideas about things that you want your course to be achieving. What a wonderful idea to actually have a way of assessing whether or not you were successful. So the reason the assessment has been so wonderful is it's told us what things we needed to change, But also, of course, what I want to brag about is that the assessment has told us that we achieved some of our basic goals. And the assessment kudos really go to Sarah Brownell and Matt Closer, who's not shown here. Sarah was a graduate student in biology, but also in the School of Education. Matt was in the School of Education. And they've now gone on to faculty positions. Sarah is going to start at University of Arizona State, okay, in January. And actually, I don't know where Matt is. Notre Dame, there you go. So we were extremely fortunate to have that team. And just to share, and you know, we're scientists, we love data, right? But this is a totally different kind of data. And this is just one set of assessments they did. They always did assessments before and after the course. And this one shows you that our students showed significant gains in self-confidence. These are all self-assessments in doing lab-related tasks, things like design your own scientific question, design your own lab protocol, interpret experimental data, present lab results to my lab members, write lab report, work as lab assistant. In every single case, students have reported gains in confidence after taking the course. But then even more exciting is that students came into the course thinking that what they were going to come away with was learning about different lab techniques. So again, here's pre and post, and you can see they have these different choices. Learn techniques, think like a scientist, design experimental experiments and analyze data. So this is before they take the course. They think that what they're going to learn about is lab techniques. But what they find afterwards is that what they really learned about was how to think like a scientist. And that's exactly what we wanted. So that's, you know, I just have carried the thrill of these results with me for years. And to share a little bit about the specific things that students said in terms of this, in answer to this question, please describe how your thinking like a scientist has changed. They learned fundamental things like experiments need to be repeated, that you need to use proper controls, both positive controls and negative controls, that you have to be critical of data. And that is going to go a long way, I think, towards informing these students for the rest of their lives, you know, long after they've forgotten about Bio44X. They learned even when the experiments didn't work. And this was really, really gratifying because this is exactly what I tell my graduate students every day, right? When you do an experiment and it works smoothly, that's great, but it's really when things don't work that gives you the opportunity to figure out new things. And then finally, that science is not a linear path. This student said, my thinking like a scientist went from straight forward thinking process to more web-like thinking process. And I think that also just really shows that the students had grasped sort of the fuller and more complex nature of scientific analysis. So I think this has been a huge success and will continue to be a success largely because of the teamwork and because we have capable hands to be handing this project off to. The last thing I just wanted to talk quickly about was I've also had a really different teaching experience, and that has been going to Africa in the summer to teach post-grad scientists there. And one of the exciting things is I've actually brought the yeast strains with me that we developed here, and they've done some of those same experiments there. And what I've really taken away from those teaching experiences, this is a very intense workshop that we teach there for 2 weeks. We're with the students basically morning, noon, and night, and we try to cram everything into them. They get lectures, they get journal clubs where we sit down and discuss research papers in detail. They get labs. We show them seminars by DVD. We just do 1,000,001 things. But I have to say that the thing that really overwhelms me is that the journal clubs, this process where you take a scientific paper, sit around in a group, and go through the paper figure by figure, line by line, this is something that our undergraduates at Stanford can do. I mean, many of them can do it as freshmen, and they can definitely do it by the time they're juniors and seniors. And yet, In Africa, the educational system and the resources available are quite different, and our students generally cannot do that, even though they've already graduated and in many cases have a master's or are doing their PhD. And this, I think, remains to be the most important thing that we really teach those students while we're there. And they don't have a lot of resources, but they have the internet, and they have the scientific literature is available to them. A lot of journals allow you to access papers for free when you're in a developing nation, but they don't have the skills. And if we can teach them the skills, that's just an amazing empowerment. This is basically the ability of anyone who wants to know the answer to any scientific question to go to the literature, download a paper, read it, but then also decide whether or not you believe the results and the interpretation. And then the other way this has just really affected me is that all of these young scientists, they're to a person. They're just really dedicated and motivated. They get no money. There's no such thing as a graduate fellowship there. Oftentimes these students, they're teaching to support themselves. They have to pay sometimes to use the bench in a particular lab. I mean, it's really, they just have a lot of hurdles, things that we take for granted here. And yet they're just so motivated because they really see this basic scientific research as a way to improve their lives and the lives of the people around them. So that's been very inspiring to me and has made me realize actually that teaching is a valuable skill, which is not always the most obvious thing when you're working at a place like Stanford. And you vie with Nobel Prize winners. So finally, I just have to say, what am I going to do next? I'm thinking about it. I'd like to follow more of the model that Tad has achieved with 44Y. I really think I would like to move on to designing lab courses for students that will also feed into my own research. So I'm starting to think about that. And take advantage of amazing colleagues in the department. Here's Professor Corney. And of course, get back in touch with my lab group. This is our traditional holiday Halloween celebration in the biology department where we do things like explode pumpkins. So thanks. Absolutely. I'm sorry I talked too long. If there aren't any questions.
Well, I guess I'll ask one. Even though you were emphasizing the value and the role of teamwork, and it seems obvious, you were also talking about environments that are much less resource endowed. So for something like the Bio 44X, is that replicable without a pretty big support structure? Or do you think there would be ways of at least creating some of that atmosphere?
I think that's a really good point. So one thing to emphasize is because that course uses a microbe, yeast, it actually is relatively inexpensive. And my sort of next goal in terms of the African scientists is to actually get some of those scientists who are now going to be teaching undergraduates there to actually do some of these experiments. So the challenge there, and it's a challenge that people face here too when they're not teaching at a research university like Stanford, but they're teaching at a small college, for example, you may not have access to a lot of lab equipment. And there are certain fundamental aspects of that course that really challenge students at the intellectual level, but really don't require a lot of fancy equipment. I mean, you need some things. You need to be able to autoclave your media, sterilize your media. And actually being in Africa makes you realize that even some of these basic things you take for granted. Like we store our yeast strains in a -80 freezer, and actually don't have -80 freezers in general, and they can't count on, they always have interruptions in the power. So that actually is a challenge. But they use liquid nitrogen to store things. So really you need to be able to make media, and you need to have some incubators to grow things at different temperatures. But
you know, it's still relatively little in terms of the equipment demands. Yeah.
Do you think some of the lessons that you've learned through teaching these college level courses would also be applicable for like the high school level, or is that, I mean, at the high school level, do you really just need to teach more facts so they can build up that knowledge base?
No, I absolutely think there's just no reason why high school students can't be doing these same things. And I think, in fact, there is a huge movement, you know, to totally revise high school and grade school curriculum along these same lines. So I haven't given a lot of credit to, you know, there are a lot of reports that have come out within the last couple of years. There's one called the Vision and Change report. I guess that actually focuses more on undergraduate level too, but all the same lessons. I mean, I think it's fundamental, and I think it goes way beyond biology too. Why do you want to be a scientist? You want to be a scientist because it's fun to figure things out. And actually, what I've seen with my kids is when they're doing chemistry labs and biology and physics labs at the high school level, it's fun because they get to do stuff, and then they go to biology and it's like, oh, it's really boring. So it doesn't have to be that way. Yeah.
Not everybody learns at the same speed. How do you handle that in your groups? Do you plan the groups ahead of time, or just start working with the problem when it arises?
You know, I think that's always a challenge. And so I certainly don't have any great answers to that. I think in the lecture course, that was always one of our biggest challenges, was that the course just goes so quickly, and you know, I just always saw students that weren't doing well, and that was frustrating. In the context, so I don't have a great answer, but that actually is one of the reasons why I really wanted to take a position at VPUE. And I think things like the Leland Scholars Program will encourage students, first of all, just to stick with it if they don't have the background, because part of the danger of students that can't keep up is that they just get discouraged and go away. So one thing is you really have to address those issues. But then also, you know, you do have to give students resources to be able to fill in the skills that they may not come with. And we have those resources here. We have a great tutoring program, right? And all kinds of, you know, supplemental courses and instructor office hours. So one of the challenges there is just making sure that students are actually taking advantage of the resources that we have. And that's a different kind of a challenge. I think also one of the things that is really a positive in the lab course is that those courses really are in small groups, and there is a lot of one-on-one instruction on the part of the graduate students, right? Sean could tell you, and the PhD level instructors with each student. And there's actually a significant amount of teaching that goes on student to student. So the fact that the students work in pairs is actually really helpful.
How did the role of the graduate students, the teaching assistants, they're in a very different kind of teaching role than they might have been in other kinds of courses. What was their role like, and was there development or a process to help them learn to teach in a different way?
Well, that's going to make me feel bad. We probably haven't done as much as we ought to. They got training in the particular experiments that were going to be done. And, geez, you know what? That's embarrassing. I don't really know what to say to that. What should I say?
Because Shamala just left,
Yeah.
unfortunately. We started, the first year we tried giving them like a 6-day boot camp the quarter before, and found out that was too far ahead. So they are actually being trained one morning a week, sort of a week ahead of the incoming lab that's happening. And there is a lot of emphasis on how to interpret data, how to ask questions of the students.
Right.
So we're trying to get it to be, no, don't answer the question, ask them a question. And I think that's coming along. And they also, each group of TAs meets with his or her lead instructor once a week to work out details on that part. And then the way we've compromised our facilities is each lab room holds 10 students, and we connect it with an open door. So a TA and an instructor is in charge of 2 rooms. So a TA is often the key person in one room and the instructor the other room, and then they alternate weeks that they're in the other room. So they get the opportunity to be in charge of their space and their students, but do the whole group
Also,
together.
just one other thing I will add is compared to the previous model, where there was usually a graduate student and an undergrad teaching each of the 2-week units. In that model, which was just an organizational feat, students rotated through, whereas the individual grad student and undergrad TA always taught the same lab module. In this model, it's also just much more contact, continuous contact through the 10 weeks with an individual instructor and an individual graduate student. And I mean, I'm sure you can give me data to cite the fact that that just works better, because you have to let relationships develop. And I think I would venture to say that the new course is also a better learning experience for our graduate students than the previous model.
And just one other thing, we do have a discussion section every week that students have to come to. So that's another way to equalize their backgrounds.
That's
because
true.
we discuss what's coming and what's happened, and they fill in there.
Besides the time they spend in the lab, they come for an additional section.
And we'll have to make that the last question, and thank you so much again.
Well, thank you, everyone,
Vice
for
Provost
coming.
Martha Cyert.